Next, the search for the genetic material. The search for genetic material. Even though the discovery of nuclein by Mischer and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as genetic material took long to be discovered and proven. Previous discoveries by Mendel, Sutton, Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. So, in this journey, there are different scientists proposed different theories. So, which molecule will be acting as a genetic material? And firstly, let us discuss about the transforming principle, which was proposed by Frederick Griffith. Transforming principle proposed by Frederick Griffith. Frederick Griffith in the year 1928, and he used the two experimental organisms. The experimental organisms are, one is Streptococcus pneumoniae bacterium, Streptococcus pneumoniae bacterium, and another one is mice. This bacterium having two strains, one is R strain, another one is S strain. R strain is nothing but the word R represents a rough strain without having polysaccharide coat, and S strain is nothing but having polysaccharide coat, which is also virulent. And this Streptococcus will be having two strains. One is R strain and another one is S strain. And R strain is avirulent or non-pathogenic. S strain is virulent or pathogenic. And when it comes to the procedure, first, Griffith used S strain bacteria. Injected into mice. And the status of the mice is die. Mice die. Next, similarly, in the second step, again he used R strain, injected into mice, but now mice is alive. Yeah. Next, instead of normal S strain, and Griffith used heat-killed S strain bacteria. He injected heat-killed S strain bacteria into mice, but mice is live. Next, in the fourth case, he injected both S strain, that is heat-killed S strain, plus live R strain bacteria. injected into mice, but again mice died. Finally, he concluded that R-strain bacteria had somehow been transformed by the heat-killed S-strain bacteria, and some transforming principle transferred from the heat-killed S-strain had enabled the R-strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiments. So that is R-strain bacteria, R-strain bacteria transformed into S-strain, R-strain bacteria transformed into S-strain. Hence, mice died.